Hi everyone, my name is Natalie Luca, and today I'm going to be sharing with you my protein structure of piezo-1. There's a lot of different parts to this, but for the sake of this video, I'm going to be describing this piezo-1 protein as if I'm describing it to my friend Amelia that is not a STEM major and she does not have any science backgrounds. I'm going to be trying to explain this in very simple terminology. However, there's a lot of different structures and parts to this, but I'm going to try to break it down the best I can to apply to you, Amelia, if you're watching this. Like I said, the protein that I'm modeling is called piezo-1. It's a mechanosensitive ion channel, which means it opens when cells experience mechanical pressure or stretching. It's located within the cell membrane, which is the outer boundary of the cell. It's on the outer portion of these cell membranes. And when that channel opens, it allows things like calcium to enter the cell and send signals to other cells. However, when there is a mutation in the piezo-1 gene, it causes things like dehydration hereditary stomatocytosis, which is a blood disorder where red blood cells lose water and become dehydrated over time, which is very bad for your body when it comes to cell mutations like that. When I'm describing my shape, I would say it has a very unique structure. These three different pieces of the structure are like a three-bladed propeller. In my model, the three straws represent the three subunits of the protein. And the cup in the middle represents the ion channel pore where the ions move through. Hence why I have two little gemstones, if you will, within my cup that represent sodium and calcium ion channels moving throughout this cell membrane or channel pore. And the cardboard, which is a paper plate for us, represents a cell membrane where the protein sits, with the outer cell membrane in this shade of blue here. When we're talking about the different protein structures, we have the primary structure. The primary structure refers to the sequence of amino acids that make up the protein in my model. The colored beads located here represent individual amino acids that are connected through each other. I used hot glue to connect these together, but it completely represents this amino acid chain. When we have the secondary structure, it holds the part of the chain that folds into shapes like the alpha helix and the beta sheets, located here. I have my alpha helix by this gold ring, and then I have this one balloon that I cut into half to make into this alpha helix, which is a common structural shape within the piezo-1 proteins. The tertiary structure, which can be described as this straw, is the overall three-dimensional folding of one protein chain. Each curved straw in my model, which these are not all straws, I wanted to make them all different to show a different type of structures within the piezo-1, but we have the three different straws, but they're just different materials, which each of them representing one fully folded piezo protein subunit. So we have the furry, the popsicle stick, and then the blue straw. The quaternary structure is the whole model in itself, meaning that multiple protein subunits come together to form a larger functional complex. In this case, the three subunits assemble to form the ion channel, as we see with the cup located within the cell membrane. The mutation, which I will describe as this black button here, that's a part of the amino acid protein chain, but as you can see, all the other buttons are colorful, with this black one cutting off the colorfulness of these different buttons that represent the amino acid chain. So that one bead is a different color, and that mutation occurs in the channel region of the protein, which can affect how easily this channel is allowed to open and close, allowing that calcium to flow in and or out of the cell membrane. Normally, piezo-1 opens only when the cell membrane experiences mechanical pressure. However, some mutations cause the channel to stay open longer than normal, and that can be we can think of that as things flowing in and out of the cup, like these gemstones here that are inside. This allows too many ions to enter the cell, which then disrupts the balance of water and ions in the red blood cells, like I talked about with the dehydrated mutation that can occur. As a result, the red blood cells, like I was talking about before, become dehydrated and damaged, which leads to that blood disorder that is associated with this piezo-1 mutation. There's multiple limitations within the model, and while my model will help visualize the structure of the piezo-1, there are many things that I'm unable to correctly show within this. The real protein is made up of 1,000 atoms, but my model simplifies the structure by using household materials and things that I found in my craft drawer. The model doesn't represent accurately the scale of size. This is large just for show, but piezo 1 is actually very, very tiny and microscopic and not something that we can see with our eyes without a different type of tool, like a microscope. It also cannot show the dynamic movement, like the movement of in and out of cells, the channel opens and closes. I was not accurately able to depict that within this model. But yeah, thank you so much for listening to my detailed description of my piezo 1. I hope this made sense to you, Amelia, and others that are listening. Like I said, it's really hard to make it simple, but I hope that this all makes sense. Thank you.